What aromatic amino acids you know? So phenylalanine, tyrosine, tryptophan. Yes? Good. So I hope that you also know the formulas. Yes? So in this lecture, we are not going to go very deep in the amino acid metabolism. Yes? In the books, the chapter about amino acids has 20 to 30 pages. There is described the fate of every amino acid in the body. Yes? This you do not have to memorize. Yes? So in the books, study especially the topics that we do today here. Yes? Of course, you can learn also the metabolic fate of all amino acids or how the amino acids are produced in our body, but it's not in the test. Yes? At the beginning, I will tell you something about the protein turnover. So how many proteins we degrade a day, how many proteins we produce a day, how the cell finds out that this protein should be degraded. And after this, we will go through the basic reactions of amino acids by transamination, by deamination, through the decarboxylation, then through the urea cycle, and then only a few words about the fate of carbon skeleton and about the synthesis of amino acids in the body. So, how many proteins a day you think we degrade? We degrade it today.
21 amino acids.
Not how many proteins, how many grams of proteins we degrade a day in our body? 100? We degraded about 400 grams of proteins a day. Yes, and the same amount we synthesize. Yes. So the protein turnover is very intense in the body. Yes. We have about 15 kilograms of proteins in the body, and we every day degraded 400 grams, and the same amount we resynthesize. The degradation and the synthesis, it means the turnover is different in different organs. For example, in skeletal muscle, we degraded about 10% of all proteins in skeletal muscle. In liver, we degraded about 40% of all proteins in liver. And in smaller intestine, it means in enterocytes, we degraded or resynthesize 80% of all proteins. Yes? So you see that the protein turnover is very, very intense. Yes? Of course, there are some organs where the protein turnover is very slow. Yes? But in small intestine, 80% of all proteins, degradation and resynthesis. Per day, we eat about 100 grams of proteins. Yes? It's different. In Western diet, we eat more. Yes? In Asia, in Africa, they eat less, of course. Yes. Do you think that it's good to eat more proteins? That's not a question whether it tastes better. Of course, it's important for if you are happy, then you will live longer. That's true. And therefore, obese people live better life, yes. But do you think that it's better to eat more proteins for our metabolism? It's not good for the problems with the nitrogen, yes. The way how to excrete the nitrogen, that's very difficult. So it's a stress for the metabolism when it has to degrade many amino acids. Normally, the same amount that you eat, the same amount you degrade it. So it means 100 to 100, and this makes about 10 to 20% of all energy expenditure. If you compare these two numbers, you gain something that is called nitrogen balance. Yes? We can have positive, neutral, or negative nitrogen balance.
How can we degrade 400 grams per day when we only need 100?
Yes, you degrade it, but these proteins that you degraded in your body, you resynthetize. Yes? So the nitrogen balance is calculated only from the amount that you take and that you oxidize. So if the amount is 100 to 100, it means you have normal, neutral nitrogen balance. Yes? In some periods in our life, we eat more proteins than we then oxidize. Yes? For example, when you are a child or when you are after some severe illness, then you store the proteins in the body for you produce new proteins. Yes? During the growth, the amino acids are used, the amino acids from diet are used to produce your own proteins. The state when you take more proteins, more amino acids than you degrade it, it's called positive nitrogen balance. Yes? So you take more nitrogen to the body than you excrete in the day. On the other hand, we have negative nitrogen balance. That's typical for stress conditions. If you have some severe illness, for example, some severe trauma due to car accident, your body starts to degrade its own proteins and you oxidize them, so you are losing the nitrogen content in the body, and this is called negative nitrogen balance. And if you survive this severe state and you start eating new amino acids, then you will develop positive nitrogen balance. Yes? So this is the positive, negative, and normally for us, neutral nitrogen balance. So here we have the scheme. We have 14 kilograms of proteins in our body. About 40 grams of proteins are degraded. The same amount are resynthesized. Here we have the proteins in the diet. And the last source of amino acids are amino acids that we can produce in the body. Per day, we can produce about 30 to 40 grams of amino acids. If we put these three together, it means the amino acids released by proteolysis, proteins in diet, they are degraded to amino acids, and the amino acids de novo synthesis, we give something that's called amino acids pool. Yes? And this amino acids pool is between 500 to 600 per day. Yes? And with this amino acid pool, we can do three things. We can produce the proteins. The majority of amino acids are used to do this. Or we can convert the amino acids to some other compounds. For example, our amino acids are used to produce nitrogen bases in DNA, to produce purines and pyrimidines, or to produce heme, or to produce the neurotransmitters, or to produce hormones. And the last thing what you can do with the amino acid pool is to degrade it. And for the degradation, it's important that there are different pathways how to degrade it. The amino nitrogen, it means the ammonia, and different fate for the carbon skeleton. Yes? So we have so many proteins for 14 kilograms, and per day in our amino acid pool we have about 500 to 600. Yes? In our body, there is no storage protein. Yes? We have triacylglycerols. They store lipids. We have glycogen. Glycogen stores glucose. But there is no protein that store amino acids. Yes, all proteins in our body have some function, and if you destroy them, you will lose this function. Yes? And therefore, the protein turnover has to be strictly regulated. Yes? For you have no reserves. How is regulated the protein turnover? Have you spoke about it? How our cell decide what it should degrade or what not? What protein? For you know, the half-life of the protein is quite different. We have enzymes for ornithine decarboxylase, and its half-life is 11 minutes. Then we have hemoglobin, and the hemoglobin has to survive the whole life of erythrocyte is about 90 to 100 days, 120 days. And then we have protein that's called gamma crystalline. That's the protein of our islands. And it has to stay during our whole life, yes? So how the cell can decide that this protein has to be there for our whole life, this also for the whole life of the erythrocyte, and this enzyme is degraded in 14 minutes, in 11 minutes. How the cell knows what protein should be destroyed, what not.
Maybe there are some signals with oligosaccharides.
There is some signal, and the signal is this protein. Have you spoken about it? Yes. So, this protein is the signal, or is the marker, is the label that you put on the protein. Yes? And the more ubiquitins the protein has, the sooner the destruction will come. Yes? So, the cell has some enzymes that are able to take this protein and to bound it to the protein molecule. And the more ubiquitins you have on the protein, the sooner it will be degraded. And the place for the degradation are proteasomes, yes? That are the organelles for protein degradation in the cell, yes? So the proteolysis in the cell is strictly regulated. It's quite complicated mechanism, for you have enzyme number 1, enzyme number 2, enzyme number 3. And that's not one enzyme, that's the whole family of enzymes. And according to the affinity of these enzymes to the substrate, it means to the proteins, it's determined the half-life of the protein. So very often the enzymes, for example, the ornithine decarboxylase, they have very high affinity to this E3 enzyme. Therefore, the ornithine decarboxylase has a lot of ubiquitin on its surface, and it's degraded in the proteasome. Yes? And the gamma crystalline has non-affinity. So it will be here the whole life. Yes? So this is the way how the proteolysis is regulated. Yes? About the regulation of proteosynthesis, you will speak in the third course about the transcription and translation. You know that in the body, we have two types of amino acids. We have essential amino acids and non-essential amino acids. And this list you should have to know, yes? Our body needs to have in the diet the essential amino acids, yes? All branched-chain amino acids are essential. So it means valine, leucine, and isoleucine are essential. 2 aromatic amino acids are essential, phenylalanine and tryptophan. Why not tyrosine? Yes, tyrosine is synthesized from phenylalanine. Yes, so it's not essential. Then one basic amino acid, that's lysine. One for sulfur, that's methionine. And one with hydroxy group, that's threonine. Here we have the list of non-essential amino acids. And somewhere in the middle, we have these two, arginine and histidine. These two amino acids are essential only sometimes in our life, yes? It means that we can produce these amino acids, but the pathways for their production have some limits. And the limits are not so high, yes? There is not a big range between the normal need or the normal amount that these acids can produce, and during some conditions we need more, yes? For example, these amino acids, arginine and histidine, are essential during the childhood, very often, yes? For during the growth, your growth, you need more amino acids, and if you do not eat these amino acids in the diet, they become essential, yes? For the pathways aren't able to produce enough arginine and histidine. And the same is true if you have patient after some severe trauma, after some severe catabolic state, and then the patient is in recovery state, the enzymes aren't able to produce enough arginine and histidine, yes? And therefore, the enteral nutrition for patients in severe state has more arginine and histidine inside, yes, to cover these body needs. And the last general topic are reactions of amino acids. So, this is amino acid. And now we can do with it 4 reactions. So, the first reaction that you can do with amino acid is to remove carboxy group. It's called decarboxylation. And if you remove this, you will gain the only functional group is this. You gain amines. And for they are produced in our body, they are called biogenic amines. Yes? Amines, for you remove this functional group with highest priority, and you have here the amino group. So it's amine. Now there are 2 possibilities to change the amino group. First possibility is that you take amino acid, some alpha keto acid, and you do transamination. In transamination, you put there the keto group, yes? So from alpha amino acid, you produce alpha keto acid. Or you do the oxidation of this amino group And during this oxidation, you remove the nitrogen, you remove the hydrogen, and incorporate oxygen. So you produce also keto group, and the result is also alpha keto acid. So the difference between transamination and oxidation is that during oxidation you need only enzyme, amino acid, you remove ammonia, you produce keto group, and you have alpha keto acid. During transamination, you only transport this amino group to other acid, and from that keto acid you transfer the keto group to the previous amino acid. Yes? So here is produced ammonia, here not. But the result is the same, alpha keto acid. And the last reaction,
Where does the O come from?
here or here? This from oxygen, that's the oxidation. There's the normal oxidation, so reaction with cofactors, as NADH. Here there comes no oxygen. There you gain the O from alpha keto acid. The last thing that you can do with these functional groups is the formation of peptide bonds. It means you let react two amino acids together. And you produce the peptide bond. About the peptide bonds, you will speak in the third course. Today we will focus only on these three reactions. Yes? Decarboxylation, transamination, oxidative deamination. So not about the peptide bond. So the first, we will take the transamination. Transamination is a very frequent reaction. It's catalyzed by enzymes that are called transaminases or aminotransferases. Yes, the same thing. We have many transaminases in the body. For one transaminase is specific only for one pair of one amino acid and one alpha keto acid. Yes? So one transaminase can catalyze only one reaction between one amino acid and one alpha keto acid. The reaction is freely reversible. Yes? So you can do reaction in one direction and also the reverse reaction. Yes? This enzyme needs to have the cofactor. In general, the metabolism of amino acids needs a lot of cofactors, a lot of vitamins. And here for this reaction you need pyridoxal phosphate. That's the derivative of vitamin B6. Here is the scheme of the reaction. Here we have one amino acid, for example, glutamate, 5 carbon atoms, and here we have some alpha keto acid. In transamination, you only exchange alpha keto, this keto group to this amino group. So from glutamate, you produce alpha keto glutarate. From alpha keto acid, you produce alpha amino acid. Yes? So no oxygen, no ammonia, no water, nothing is produced, only exchange. You should learn these two transaminases, for they are very important for the human metabolism. The first is alanine aminotransferase, ALT, and the second is aspartate aminotransferase, AST. Alanine aminotransferase takes alanine, that's the amino acid, takes alpha ketoglutarate, that's the alpha keto acid, and the product is pyruvate and glutamate. Or the possibility is to take glutamate and pyruvate and to produce alanine and alpha ketoglutarate. Yes? Aspartate aminotransferase takes aspartate and alpha ketoglutarate and produces oxaloacetate and glutamate. This is one example of anaplerotic reaction in Krebs cycle. Yes, you take you take amino acid, and you produce oxaloacetate, intermediate of Krebs cycle. So this is transamination. Simple reaction, only exchange of amino and keto group. Second are conversion glutamate and glutamine. You know, metabolism of alpha ketoglutarate, glutamate, and glutamine is very intense in the body. Yes, glutamate, alpha ketoglutarate can be produced easily, and in glutamate and glutamine you can store the amino groups. Yes, in glutamate you have one amino group, and in glutamine you have 2 molecules of ammonia. Yes. Conversion of these 2 amino acids is quite simple. It's the conversion of this carboxy group in glutamate to this amide group in glutamine. Yes? And then the reverse change of this amide group to carboxy group. The first reaction, it means from glutamate to glutamine, is catalyzed by glutamine synthetase. Yes, by cytosolic enzyme, glutamine synthetase. And when it's called synthetase, it needs ATP. Yes? For this reaction, you need ammonia, free ammonia. Yes? And this free ammonia is incorporated here to the amide group. The glutamine is the form how our body transports ammonia through our body. The problem is that pure ammonia is toxic for us. Yes? So you need to put it into some untoxic compound. Yes? Ammonia is produced in our whole body and is toxic. So our peripheral tissues take this ammonia and produce glutamine from it. From glutamine is untoxic amino acid. And then this glutamine, therefore the glutamine has the highest concentration of all amino acids in blood. Yes? For it's the transport form of ammonia. And then this glutamine is transported to liver mitochondria. Why to liver mitochondria? For liver mitochondria, that's the place where you have the urea cycle. Yes? That's the way how the ammonia is detoxified in our body. You produce untoxic urea. And then in liver mitochondria, you have the enzyme that's called glutaminase. It's a simple hydrolysis that takes water and destroys this amide bond and produces the glutamate. Yes? So these reactions are opposite. Important is that they are catalyzed in different cell compartments and also in different organs. Yes? This is typical reaction for all our body. Yes? Is the way how we conserve ammonia to some untoxic compound. And then in liver mitochondria, we releases the toxic ammonia from glutamine, and this toxic ammonia is detoxified in urea cycle. You understand this conversion? This is in cytosol, and in the whole body it can be. In the whole body. All cells, not liver, yes, for liver do not need it, yes. But the glutamine synthetase reaction is in the whole body, yes. We call in extrahepatic tissues. And it's in cytosol. Oxidative deamination of glutamate. It means this oxidation and you release ammonia. You know, the deamination you can do with all amino acids, but only the deamination of glutamate is important, yes? For the activity of other enzymes is very, very small. Oxidative deamination of glutamate is catalyzed by glutamate dehydrogenase. Dehydrogenase, why there is oxidation. This reaction is also localized in liver mitochondria especially. Why in liver mitochondria? For the product of this reaction is ammonia, and only in liver you can get rid of ammonia to produce urea. This glutamate, that was this glutamate, yes? So in liver, you do two reactions. The first reaction you remove first ammonia from glutamine to glutamate, and then you take this glutamate and you do the oxidative decarboxylation. Yes? So there are two steps in the liver that you do. The product of this reaction is alpha-ketoglutarate. So glutamate, alpha-ketoglutarate, and ammonia. The ammonia goes to urea cycle, and alpha-ketoglutarate can decide how the liver mitochondria will use it. From alpha-ketoglutarate, it can go to the urea cycle, it can produce glucose, or it can go to another transamination. So, if we put this together, then if you have some amino acid that you want to degrade, yes, for example, alanine you want to degrade, in the first step, you do the transamination and you put the ammonia or the amino group from alanine to alpha-ketoglutarate and produce the glutamate. And then in the liver, you take the glutamate and you release the ammonia from it. Do you understand this? Yes? If you have some amino acid that you want to degrade, the first step is the transamination to take this amino group to alpha-ketoglutarate. You produce glutamate. And then in liver, you do the oxidative deamination, where you releases the ammonia. And the ammonia is detoxified in the urea cycle. And if you have excess of ammonia, then you will convert glutamate to glutamine by glutamine synthetase. And then in the liver, you use also the glutaminase. Is it clear? So this is written here. So in extrahepatic tissue, if you want to degrade amino acid, you have possibility to do the transamination and amidation of glutamate. And then in the liver, you releases the ammonia by glutaminase and by oxidative deamination of glutamate. So this is the cycle of ammonia. Yes? Want to degrade amino acid in peripheral tissues, so you do the transamination and amidation of glutamate to produce glutamine, and then in liver you do the reverse reactions. Urea cycle. Urea cycle, that's the way how our body detoxifies the toxic ammonia. Ammonia is toxic for all cells, but especially it's toxic for brain. Why you think for brain?
It changes the pH.
That's also true. It changes the pH as a base.
It can pass through the blood-brain barrier.
Yes, it can go through blood-brain barrier, that's true. But why it's more toxic for brain than, for example, for bones? Both begins with B. Or blood cells.
Because it may signal something wrong with the brain.
That's also true, but not itself ammonia can do the signal. But some compound that it produces is the signal.
So it can create neurotransmitters.
Yes, but how it can produce this? The production of neurotransmitters, that's true, but in brain and why not in bones? For in brain you have very intense energy metabolism, yes? That's together with liver, brain, these two organs has very intense energy metabolism. And the problem of ammonia is that ammonia is in many reactions, yes? For example, you have alpha-ketoglutarate and glutamate. Alpha-ketoglutarate is in Krebs cycle, and if you have a lot of ammonia together with a lot of alpha-ketoglutarate, you will produce glutamate. So you will take the intermediates of Krebs cycle out and you will block the Krebs cycle. Another true thing is that glutamate is a neurotransmitter, so also high concentration of ammonia will change the neurotransmission. You have more excitatory signals in the brain. Yes, we call it glutamate excitotoxicity. Yes. So therefore, it's so toxic for the brain. For the brain needs a lot of energy, and if you start to drain alpha-ketoglutarate from the Krebs cycle, you impair the energy production, and also the products, as for example, glutamate or aspartate, they are neurotransmitters, so it can change the brain function.
Do you differ between ammonia and ammonium?
Yes, one is NH3 and the second is NH4+.
Yeah, but
When I said this, I mean the same thing.
okay.
Yes, absolutely the same. Ammonia and ammonium, there is difference, but
Yeah.
you know, there is the balance between ammonia and ammonium in the body that is dependent on the pH. And therefore, our body keeps the concentration so low, below 35 micromoles. And the concentration of glutamine is about 2 millimoles. And the only function of glutamine in the blood is to store the ammonia. So without glutamine, we are dead. Yes? For the concentration of ammonia, 2 millimoles, you cannot survive. If the patient has concentration about 130/40, the patient is unconscious. Yes? The brain stops working. So the urea cycle is the way how to detoxify the ammonia. 99% of urea cycle is localized in liver. Yes? In the books, you will read that urea cycle is localized only in liver. Unfortunately, about 3 years ago, some student came with an article that he found that in kidneys is 0.5% activity, and this way, yes? So therefore, we said that about 99% of urea cycle in liver, the rest in the body, yes? But if you remove the liver, you will die. So it's important, and the urea is excreted in kidneys, yes? So urea produced in liver, excreted especially in kidneys. Do you know the formula of urea? So can you help me? So how many carbon atoms it has? So this is urea. So if you want to produce urea, you need 1 carbon dioxide and 2 ammonias. The thing is that you need only 1 ammonia in form of ammonia, and you will use 1 amino acid as a donor of ammonia. Yes, it will be aspartate. Yes, and you will change aspartate to oxaloacetate. Yes, so for urea production, you need ammonia, carbon dioxide, and aspartate. This reaction is localized in 2 compartments. In mitochondria, 1 part, the beginning, and some reactions in cytosol. The regulatory enzyme is the first enzyme of urea production. Is called carbamoyl phosphate synthetase number 1. Why number 1? This carbamoyl phosphate synthetase is in mitochondria. It's important for the urea production. Then we have carbamoyl phosphate synthetase number 2. It's the cytosolic enzyme, and it's important for production of pyrimidines. Yes, about the carbamoyl phosphate synthetase number 2 you will speak in the next course. The whole cycle needs a lot of energy. Yes, therefore it's localized in liver, for liver has still a lot of energy. And it's closely connected with the Krebs cycle. The part that's in mitochondria is closely connected with Krebs cycle. So, first reaction. That's the reaction catalyzed by carbamoyl phosphate synthetase, number 1. That's the reaction that's localized in mitochondria. And the reaction is quite simple. You combine carbon dioxide, or bicarbonate, that's the same thing. Or also there is the balance between carbon dioxide, carbonic acid, and bicarbonate. And it costs you 2 ATPs. You see that there are many steps. For you, the steps between aren't important. Only know that you have ammonium, bicarbonate, 2 ATPs, and you produce the carbamoyl phosphate. Carbamoyl phosphate is when you remove this and put there the phosphate group. Yes, that's carbamoyl phosphate. So 1 ATP puts the phosphate inside. The other ATP is only important to give energy to the reaction. Yes? Then we have the second reaction. We have the carbamoyl phosphate, and then we have this amino acid, ornithine. You can see that this is amino acid, yes? But this amino acid you cannot find in our proteins, yes? So ornithine is an example of alpha amino acid that you cannot find in our proteins, but it's important for our metabolism, yes? Therefore, if you will read in the text question, in our body we have 20 alpha amino acids. Is it correct or not? Not. We have, for example, ornithine. If the question is this, if the statement is, in our proteins we have 20 amino acids, is it true or not? True. Also not true. Also not 21. Yes, in the test now we have to put 20 in brackets 21. Yes, but it's also not true. For amino acids in proteins can be transformed. They can be, in the proteins you can have derivatives of amino acids. For example, hydroxyproline or hydroxylysine. So the correct statement is, during translation, you can incorporate in proteins 20 amino acids. That's true. Or 20, 21. Yes? So in our body we have many, many amino acids. Yes? Some of them are alpha amino acids, and only 20 of them can be incorporated into proteins on ribosomes. But then these proteins can be changed. Yes? So only for you this is important. Yes? For very often these statements are in the test. Yes? In our body we have 20 alpha amino acids. Incorrect. In our proteins we have 20 amino acids. Also incorrect. But into the proteins during transcription you can give only 20 amino acids. That's correct. Yes? So this is one example of amino acid that you cannot find in proteins. Yes? And it combines together with carbamoyl phosphate. You know, these enzymes you do not have to remember absolutely. Yes? Also the formulas of ornithine, it's not necessary you to know. Yes? It helps you to understand the process, how it works. But in the test there is no question how many carbon atoms has ornithine. Yes? And you produce citrulline. Citrulline is other alpha amino acid. You cannot find it in proteins, but it's very important for the amino acids metabolism. The process is in this reaction that you remove the phosphate, and this carbon atom binds to this nitrogen atom. And here you have it. Together, it's called citrulline. And here you can see the urea. Yes? Carbon, double bond, nitrogen, nitrogen. So now all of us would accept that will come some enzyme that will do this. Yes? It will be nice, but it's not true. For we have other reactions. For we take citrulline and we use now the aspartate. So the second donor of ammonia to urea cycle. Here we have the amino group. And these two compounds combine together and produce one amino acid that you know. This is arginine. And here we have dicarboxylic acid with 4 carbon atoms. That's succinate. And therefore this compound is called arginosuccinate. Yes, this is the formula of succinate. This formula you have to know. And here you have succinate. Yes? So in this reaction, this nitrogen combines here. For this reaction, you need 2 more macroallergic bonds. Yes? So at the beginning was 2, now other 2. And you produce arginosuccinate. And now if you look at this, so here is the ammonia, here is the urea. Yes? The red and the blue nitrogen, this carbon, this will give us urea. So this nitrogen stays the whole life in the urea cycle. Yes? These 2 ammonias are removed as a urea. And now only 2 reactions when we degrade it and when we release the ammonia, the urea. Arginosuccinate, here we do the hydrolysis and we produce arginine and fumarate. Fumarate is dicarboxylic acid with 4 carbon atoms and 1 double bond. And what's the conformation? This trans, yes? Maleic acid is with cis conformation. And the last reaction of urea cycle, arginine, other hydrolysis, and here you gain the urea that you want, and you recover ornithine. And ornithine was in the second reaction when ornithine plus carbamoyl phosphate. So now we close the urea cycle. And this ornithine is transported back to the mitochondria. So this was the whole urea cycle. Do you want to move one time through it? Or do you understand? Or before question?
I was just wondering, like, we don't need to know all the enzymes and compounds?
Not the enzymes, not only the first regulatory enzyme. And the compounds, of course, you do not have to know. For example, citrulline and ornithine, that's true, but arginine you have to know for its amino acid. Yes? So the only intermediate that we can ask you is this and this. So now we have the urea produced in liver and excreted in kidneys. And before the break, I will show you how urea cycle is connected to Krebs cycle. The connection is quite simple. This is urea cycle. Here we have Krebs cycle. One ammonia goes direct to urea cycle. One ammonia goes in form of aspartate. And product of urea cycle is fumarate. So this fumarate has to become aspartate. Yes? This fumarate, the product of urea cycle, goes to Krebs cycle. From fumarate, you do addition of water, produce malate. Then you do the oxidation of malate to oxaloacetate. And then, by transamination, you change oxaloacetate to aspartate. How is called the enzyme that does this transamination? You have to remember only 2 transaminases. Alanine aminotransferase and aspartate aminotransferase. And now we will use the aspartate aminotransferase. So here, the enzyme is called aspartate aminotransferase. So the oxaloacetate is transaminated to aspartate. The donor of amino group is glutamate. So this alpha amino acid is glutamate, and this alpha keto acid is alpha ketoglutarate. And this aspartate can go to the urea cycle. You produce fumarate. And this purple part, that's the part of the Krebs cycle that you will use. Yes? Fumarate, malate, oxaloacetate. How it's regulated, the urea cycle. The regulation is quite difficult, at least this part. Therefore, we will start with this. So, urea cycle is the way how the body gets rid of ammonia. Ammonia is produced in the body when we have there a lot of amino acids, and we have a lot of amino acids when we take a lot of proteins. Yes? It means the more proteins you eat, the more enzymes of urea cycle you need. Yes? And it means that some amino acids serve as a transcriptional factor that upregulates the transcription of urea cycle enzymes. Yes? It means some amino acids can accelerate the production of urea enzymes. It means the more proteins you eat, the more enzymes you produce. Other regulatory effect. Urea cycle is an example of proton productive reaction. Proton productive reaction means that during this pathway, you produce some hydrogen protons. Where in urea cycle you produce the hydrogen protons? I will help you. So, here, do we produce?
No.
Yes. No. Here? No. Here? 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 So where we produce the protons? Maybe the pictures are bad. That's the first possibility. Maybe I'm stupid. That's also one possibility. The second meaning of proton productive reaction is that you remove some base. For normally you have balance, ammonia and ammonium. It means ammonia, ammonium. This is the base, this is the conjugated. And this base normally from the environment takes the protons. Yes, it takes it, consume it, and produce the conjugate. If you remove this, here are the protons. Yes, so in the urea cycle, you do not produce any acid, but from the body you remove bases. So it's the same, yes? Proton productive reaction is production of hydrogen protons or removing of bases, yes? What other example of proton productive pathway you know? And truly proton productive reaction where you produce acid.
Electron transport chain.
Where? Where in electron transport chain?
1, 3, and 4. Complex 1, 3, and 4.
What's the product of electron transport chain? The hydrogen gradient, but it's only the transfer of hydrogen and water. There is no acid. And only then you combine phosphates together. But for example, glycolysis. Glycolysis, at the beginning you have glucose, neutral molecule, and at the end you have pyruvate or lactate. Yes? 2 acids. So glycolysis is an example of proton productive reaction where you produce the acids. But back to urea cycle, for it leads to acidosis, normally, yes. It removes bases, and therefore it has to be inhibited. When the pH is very, very low, you have to stop the urea cycle, for that can kill you. And now, about the pure regulation, carbamoyl phosphate synthetase is a regulatory enzyme. And the regulation is quite difficult. For the allosteric activator is called N-acetyl glutamate. This N-acetyl glutamate is produced from acetyl coenzyme A and glutamate, and this reaction is catalyzed by N-acetyl glutamate synthetase. This you can skip, but what is important that arginine, one amino acid, activates this enzyme. And therefore you can say that some amino acids are activators of carbamoyl phosphate synthetase. It's not direct, but indirect there are. Yes? So amino acids are activators for transcription, and also they are activators of the regulatory enzyme. So, now 3 short topics. For every topic we have 5 minutes. So, first, utilization of carbon skeleton. From the biochemical point of view, in amino acid we have 2 things, amino group and the rest we call carbon skeleton. Yes? Before the break, you saw how we can get rid of the amino group and how to produce urea. Now what we can do with carbon skeleton, each carbon skeleton is converted by the original pathway. Some are difficult, some not. For you it's good that you do not have to memorize them. In the books you will read them. What you have to know is that all carbon skeletons can be converted to 7 intermediates. The intermediates you have to know. It can be acetyl coenzyme A, or it can be acetoacetyl coenzyme A. Or it can be pyruvate, alpha-ketoglutarate, succinyl coenzyme A, fumarate, and oxaloacetate. All these 4 things are intermediates of Krebs cycle. So we can simplify it more that amino acids can be degraded to acetyl coenzyme A, to acetoacetyl coenzyme A, to pyruvate, or to intermediates of Krebs cycle. And depending on this, we can divide our amino acids into 2 groups. To ketogenic amino acids and to glucogenic amino acids. If amino acids is degraded to these compounds, you can produce from that amino acid glucose. Then it's glucogenic. If the amino acid is degraded to acetyl coenzyme A or to acetoacetyl coenzyme A, you cannot gain glucose from it, but only lipids. Therefore, they are called ketogenic amino acids. And some amino acids give you this and also this, for they are big. For example, tryptophan is big, and it can give you both intermediates. Then it's glucogenic and ketogenic, or glucoketogenic. Here you have the list. For you, it's important to learn only this. There are only 2 pure ketogenic amino acids. Those that begin with L, it means lysine and leucine. Or from all other amino acids, you can gain glucose. Yes? So remember 2 ketogenic amino acids, lysine and leucine. In the books, you will find these nice schemes. Yes? Last year, I had there 4 pictures from 4 different books. And we try to find there the differences. Yes? For in different books, you have very often different scheme. You can learn it if you want, then you will be more clever than your teachers. Maybe only Dr. Trnka knows all of this, yes? And then you will look like this way. What is for us important, it means for what your teachers knows. Now I'm speaking about myself, yes? For Professor Samsova also knows everything from this. But what is easy to deduce and what's not difficult. And this you have to know, yes? Not the whole scheme, but this you have to know. Some amino acids are very easy to degrade. Can be easily transferred to, for example, intermediates of Krebs cycle. aspartate, asparagine, 4 carbon atoms transformed to oxaloacetate by transamination. The name of the enzyme is aspartate aminotransferase. Glutamine, glutamate are changed to alpha-ketoglutarate. You have to know the enzymes, glutaminase and then transaminases. Alanine, 3 carbon atoms, pyruvate, also 3 carbon atoms. Enzyme is called alanine aminotransferase. Lysine and leucine are ketogenic, so you know that they have to be degraded to acetyl coenzyme A and acetoacetyl coenzyme A. And then these 3 small amino acids, it means glycine, serine, and cysteine, they are converted to pyruvate, the same thing as alanine. Yes? So this you have to know. And the enzymes you have to know for these 3. Yes?
Can you write the enzymes for the PowerPoint? Can you put the enzymes into the PowerPoint?
The enzymes are in the PowerPoint on the previous slides. Here you have the glutaminase, that's there. Or glutamate dehydrogenase, that's also there. Here the transamination with aspartate, that's aspartate aminotransferase. And this alanine aminotransferase.